Okay. Okay. Who's freaked out for this exam?
I already took it.
I met with a former student of mine the other day, and she was like, "People are saying that you're hard," and I'm like, "How am I any different than I don't know? Than people who were in cell biology are saying that I'm hard in this class. Do you guys think that's true? Is it any different?"
It's pretty similar.
Okay. I don't think I'm a hard teacher. Quizzes are supposed to be hard because they're open book. And you can call your friends. You can call Nobel
We're
Laureate.
supposed to call our friends?
Yeah.
That are we completed?
Okay, so if someone didn't realize you can call a friend to do a quiz. You can call a friend. You can sit together and take the whole quiz together. Okay. So this is going to be a fairly short lecture because I went to go adapt the lecture from last year and realized it was a guest speaker and it was completely not my style and blah-de blah. So I had to do it from scratch with very little time. And I figured, you guys will be happy because you get to go eat lunch and then take an exam. So it's fine. Exam 1 this week, you already know that. Don't discuss the exams, please. And the gene expression topics are due in recitation next week. Those who were not here on Monday, talk to your friends because I introduced the project. It's also on the lecture from, which I probably haven't uploaded yet, so I'm sorry. Probably from the lecture, on the lecture from last Monday. Okay. Does anyone, before we get started, does anyone have questions that they've run into already about with the gene expression project that you would like me to clarify? And if it's explain the project, then that's not one that I can do right now. Yes?
What if you're one of your group members not coming to lecture? How will contact with them?
If one of your group members has not come to lecture, do you have their contact information? Ideally, in the beginning of a semester, you exchange contact information with your group members to talk to your other group members and see if they have the contact information, and then email me if you still can't get in touch with someone.
Okay.
Okay. Any other questions about what you're doing, what I expect of you next week? I really expect that you'll have chosen your genes and that you have a reason why. Like you've actually done a little bit of background thinking. Okay? Okay. Okay. So we've been talking about transcription and gene expression. And so I modified the slide that I used last week to make it a little more accurate, right? So reading the genome, the process of gene expression, that's copying your whatever gene of interest from your DNA to RNA, and then making it into protein if it's a coding gene, or another RNA if it's a non-coding RNA. So I want you to keep those straight in your head, that you can have coding RNAs that make proteins and non-coding RNAs that do other things. Okay? And another concept that's important to understand and we can think about for a minute. This kind of relates to the question we had the other day about constitutively active genes and what makes them constitutively active versus other levels of expression. So you can have different genes that are transcribed with different efficiencies. So what this is showing is two genes, A and B, and you might get a lot of RNA when you make one. Who thought you only make one copy of RNA? Okay, thank you for admitting that. You can make one copy or you can make many copies depending on how much protein you're going to need or how many RNAs you're going to need, depending on the function. So everything is regulated. Yes?
So many copies of the same RNA?
Oh, yeah. Yep, absolutely, yes.
So when you copy your RNA, I mean, it has to go one at a time.
Does it? It's all dependent upon the number of RNA polymerases that bind.
I mean, like, you can't,
You're going to make one RNA per polymerase
right.
at one time. But you can have Okay, well, I'm going to show you a picture in a minute, which is going to show you how you can have many RNA polymerases bound to one gene, transcribing and making many RNAs. And that's part of when we're talking about real-time PCR, which we are going to be thinking about very deeply. We're talking about copy number, how many copies of RNA are present in the cell in response to, in the case of our project, viral infection, because that's a sign of the response to whatever, to the virus in our case, right? So if you're just making one RNA, is real-time PCR useful? No, because you're quantifying how much RNA, you're quantifying the level of gene expression by counting how many RNAs, and that says something about how highly the gene is expressed, right? So you can have gene expression, you can have transcription at different levels. Here you're getting a lot of RNA, and here you're just getting one, and then this is going to result in a lot more protein than that one, right? So why does this matter? Why am I telling you about this? When would a cell want to do scenario A? Yeah.
But it's upregulating a gene because of the signal that calls for the need of expression.
Well, it's upregulating a gene because there's some kind of signal that's calling for needing expression. Do you want to add? Huh? Development or growth. So if you need a lot of some protein or some RNA that's involved with shutting something down, or yep. What else? Okay, so those are two good ones. So if you need a lot of something, you're going to make a lot of something, right? And then gene B, very low levels. Why would you want to make very low levels of RNA? Let's try someone else. Yes.
Just like normal cell functions.
Normal cell functions, maybe. So maybe you don't need very high expression of a certain gene. You just need limited amounts. What else? Why might you want to limit the amount of RNA? Yeah.
Maybe you don't need the protein at that time, but you need it to be around.
So maybe you don't need. So maybe you're not inducing expression of high levels of a protein, but you still need some. Maybe. So what else? I forgot my list.
Depending on what the protein is, you worry about toxicity levels. You don't want too much in your cell.
Okay, so maybe you don't want too much of a protein, and you're going to limit how much there can be, either due to toxicity or just in terms of regulating certain processes, right? Just like you might purposefully degrade a protein if you don't want it anymore, you might also make low levels of it if you want to really tightly regulate how much of it's going to be active. So you can regulate that at the transcriptional level as well as at the protein stability level. Yeah. Did you want to add?
So I was gonna say that if you don't need it for a long amount of time, you have to.
Yeah. Did you want to add something else?
So something like a baseline process where you need a certain level at all times, which can be interactive.
Okay, so yeah, so sometimes you might need a consistent level, but then it depends on what level that is, how much RNA you're going to make. Right, so this is an important concept to understand because I think when we learn about this the first time, we think of it as replication, you're copying your DNA, one copy of DNA. Transcription, you're copying your DNA into RNA, one copy of RNA, and it's not that. It's totally different. Okay, any questions about this concept? It's a good one to understand because you can't really understand evaluating gene expression if you don't understand this yet. It has nothing to do with where the genes are. This is just the artist's version of it. Here they're showing two that are next to each other so you can easily compare and contrast, but there's no relationship between the two. Every gene has its own, every gene is, the level at which it will be expressed is specifically regulated based on that gene and the signal that turns that gene on. So it could be really far away. Yeah. Okay, any other questions? Does everyone feel good? Okay. Okay. So let's just talk about DNA and RNA for a little bit. I kind of feel like I didn't need to do this, but I did it anyways because sometimes you take things for granted the first few times you hear it, and then you think about it more deeply as you understand things more. Okay, so 3 structural differences between DNA and RNA. So can you guys, I have 2 of them up here, but who wants to tell me some structural differences between DNA and RNA? Yes?
DNA obviously has one less oxygen, and it is antiparallel when it takes its structure, while RNA it is single-stranded, so it loops around on itself.
Okay, so one less oxygen in DNA. What's that called?
That's deoxyribose.
Huh?
Deoxyribose.
Deoxyribose. And RNA has what?
Yes, ribose.
Ribose, right? So the sugar on the bases, right? The structure. So I heard something about anti-parallel helix with DNA. What's the structure of an RNA? It's single-stranded, and it does what? I heard it over there. So I heard hairpins. What else? Structure, secondary structure, right? Did anyone know that there was a secondary structure to RNA? Who loves that idea? You know, it's much easier to predict the secondary structure of RNA than of protein. Just saying. I've tried to predict structures of protein. That's a mess. Yes?
I just have a quick question. All right, I just have a question. I'm not sure, maybe I just don't understand, but you know when you have the RNA and it's the same, it's a gene that's making multiple RNAs. Is it making multiple RNAs in the same? It's just the same copy. How does it just reset, or what is that? You know what I mean?
So when you have a gene and it's making multiple copies of RNA, you're getting multiple copies of RNA. How does that happen? That's your question.
Yeah.
Lots of RNA polymerases.
Okay, so like a new RNA polymerase comes in and
Yep.
it just kind of like keeps going.
Yep, and you're gonna see a picture of it in a minute, right? Yeah. So if you want to visualize it, you can imagine, you know, your RNA polymerase. It recognizes its transcriptional start site and transcribes, right? And it gets out of the way of that start site, and that's available for another RNA polymerase to bind. And you can think of it as like one after the other in a line. Lots of RNA polymerases transcribing one after the other. Yep. Okay, so I heard 2 differences. One was the structure, and one was the sugar, deoxyribose, and ribose. What's one more? Let's try someone else. Trudy May. Base. Okay, base. What base is different in RNA? Uracil. Okay, right. So instead of thymine, DNA, you have uracil. So you guys all knew this all. The reason I put it in here is it's sometimes nice to kind of look at structures and remember, right? So this is the where the sugar differs in this oxygen. You've got the oxygen in RNA and you don't. So deoxy, no oxy, right? And then the uracil and the thymine, and they can both still base pair with A, right? Okay. So that's just what I told you. Sugars are different. One base is different. So what is this? RNA, right? How can you tell? Well, there's a uracil which helps, yeah. But what? Let's say the uracil isn't there. How can you tell? Right, there's no OH, not just an H, right? That pink OH. Okay, I might put something like this on an exam. It could be fun. It's a nice, easy giveaway question, and it allows for me to put a picture on the exam. Pictures are fun. Pictures are fun. Okay, and so this is what we're talking about with structure, right? So you know what a double helix looks like, right? Am I always blocking your way? I kind of feel like I'm always like. Okay, you have, I guess you can move. Okay. I always want to stay near the computer so you can hear me. Right, so different structures, right? So there's actual base pairing within that RNA. It kind of folds back on itself. So whenever you've got complementary sequences, it forms these hairpins and whatnot. Yes?
What are the out, like, you see how the branch backwards?
Yeah, these.
What is that?
Yeah, I didn't realize that could happen, but I guess it can, where you can have.
So it's just out base pair? Like it's just
Yeah, so something about the structure allows them to, and that kind of formed the secondary structure, right? So This is, so sometimes I've got people in my lab and they're asking why PCR isn't working. And I say to them, well, you might have designed bad primers. And they're like, what do you mean? Like, because you think of primers as well, as long as they bind to the sequence of the DNA that you're trying to amplify, aren't they good primers? Well, if you design a primer that kind of does this, it'll never bind to what you're trying to amplify. It'll just bind to itself, and that will cause a big mess. And so if you're ever in a place where you're designing primers, there are prediction programs to make sure you can actually determine whether or not this is going to happen with your primers and design them so that they don't fold back on each other. Yes?
So these are just kind of random associations. Like say if you were to copy the same RNA sequence 100 times, you can get the exact same folding.
So the question is, these are random associations. You wouldn't get the exact same folding.
Not random as like a non-base pairing, but random as their overall structure as they.
So I guess the question I have for you, so the question is, what determines the secondary structure of RNA? That's the real question, right? So what determines the secondary structure of RNA? Who wants to take a guess? How about someone in the back row? Who in the back row feels like answering this question? What determines the secondary structure of RNA? The same answer is what determines the secondary structure of protein. Yes?
How the nucleic acids are interacting.
How the nucleic acids interact with each other, which is based on the sequence of the RNA. So the same RNA is always going to have the same sequence, and so if they're close enough, they are going to want to fold back on each other, right? Or close enough, or not too close. So for example, just based on the structure of RNA, you're never going to have base pairing here just because they can't get close enough because of how they're folding. But they'll probably always, if you've got a long enough string of them that are complementary, that will always want to fold back. And that's kind of how microRNAs work, or the RNAs involved in splicing. They all have to have the same structure all the time. Otherwise, they wouldn't work. Yeah.
I just think statistically, what is to say one sequence of AA, GG matches up with the correct sequence of GG? Can't you fold? Isn't there a possibility where you can fold in multiple ways?
So the question is but can't you fold an RNA in multiple ways? It all has a different base pairing. It's going to have different energies, and it's going to always want to fold to the most favorable energy, and that is dependent upon those interactions. Yes?
So is it possible for an RNA to fold and form a double helix?
Is it possible for an RNA to fold and form a double helix? I don't know. Maybe if you've got, so you're thinking, if you've got a long RNA that's completely complementary, opposite, like a palindrome on opposite sides, could it come together like a double helix? Maybe, why not? That's, I think, within reason. Yeah. Okay, any other questions about this? I once had to sit through a whole lecture on RNA secondary structure when I was taking molecular biology in grad school, because the person who gave the lecture worked on RNA secondary structure. Whole entire, like an hour, a whole hour of structures. Got really boring actually. But anyways. Okay, so how is this similar to protein? That's because remember the protein amino acid sequence is what determines the structure of a protein, right? So you can say the same thing about folding of a protein. And that's kind of what happens when proteins are folding, is they're constantly kind of randomly coming together until they reach the most favorable energy, right? Not randomly and with the help of chaperones. Yeah. Depends. Okay. Any other questions? Any thoughts? Good. Okay. So RNA polymerization occurs 5' to 3', and the nucleotides are added in the order of DNA sequence. You knew this. Figure I should put it there, just in case someone forgot. So transcription has similarities and differences to the process of DNA replication. So there are some similarities. So just like with DNA replication, the sequence is determined by base pairing. And synthesis occurs 5' to 3'. But what's different is the RNA does not stay hydrogen bonded to the DNA template strand, right? That's what happens with DNA replication, but with transcription, the RNA is actually displaced. So here's your double helix. It gets opened up, and you have polymerization, and then it kind of comes out. It gets displaced, so it's not able to. It's kind of being unzipped as the polymerase moves along. You can think of it that way, and it comes out. There's There's a exit channel where it actually leaves. So it's not staying base paired like in DNA replication. And the RNA produced from transcription is much shorter than the DNA produced from replication, right? Right? Who believes that? Who wants to argue with me about that? Why is the RNA produced from transcription shorter than the DNA produced from replication? It's an obvious one. You guys are thinking right now, wait, how could she be asking me such an easy question? Yep.
Well, with transcription, you're only transcribing one gene, whereas with replication, you're duplicating the entire genome.
Right, so with transcription, you're transcribing one gene. With replication, you're duplicating the entire genome. Do you want to answer that?
And also you're slicing up the
Depending on your species, sometimes you are splicing out introns. Are you always splicing out introns? What's a situation where you're not splicing out introns? Who splices? Who has splicing machinery? Yeah. Eukaryotes, right? In bacteria, you don't have splicing. Yeah.
Would alternative splicing be done
Alternative splicing is a situation where different introns are removed. Yep. Okay. Good. This is a picture I was talking about. So this is two genes being transcribed. So that's your DNA. And each thing coming off is an RNA. Okay?
They seem to ball up at the end. Is that then forming a secondary structure?
Probably. So why are they balling up at the end? Probably forming secondary structure. Let me think for a minute. Because RNA polymerase is here, and the RNA is exiting out the exit channel. That could be the only thing it could be. I have no idea. It could also be an artifact. This is electron microscopy, and they do it by doing, they put like these metal shavings on the whatever they're trying to take a picture of. So it could be just an artifact. I'm not sure. Yep. So everyone sees there's these little balls at the end. So that was the question. What are these little balls? But you can see there's a lot of RNAs coming from each of these genes, right? I guess if you wanted to, you could count them. And so this is where it's starting and this is where it's ending, right? So there's a very small amount of transcript made here, whereas they're much longer over here, right? See that? Pretty amazing, huh? Amazing that they can do that. So the rate of nucleotide addition, just some fun facts. I like these fun facts. You don't have to spit this back at me on the exam. If you do, I'll actually think less of you, probably. Rate of nucleotide addition, about 20 nucleotides per second. That's fast, huh? Did you say that's fast? And so you can actually get transcription of more than 1,000 transcripts from one gene in an hour. That's a fast response time, right? Is that good? That kind of makes a lot of sense when you think about how the cell needs to constantly be adapting to changing environments. And you want to quickly respond to, if you have low oxygen and you hang around for a really long time in a hypoxic environment, are you going to survive as a species? Probably not. But if you rapidly increase expression of genes that are going to allow you to adapt to low oxygen, probably have a selective advantage. Yes?
Okay, I just wonder, how does RNA polymerase fix their errors?
Huh?
How does RNA polymerase
Okay, we'll get that in 1 minute. How does RNA polymerase fix their errors? Okay, I think that might be the next. Were you peeking at my slides? Yeah, so that's coming right up. Okay, so let's talk about RNA polymerase and compare it to DNA polymerase. The structure is completely different. So remember we talked about DNA polymerase and its structure and how it was similar to telomerase, right? And it's similar to some viral polymerases. It's absolutely, positively different from RNA polymerase. And that has a lot to do with the function, but also it's just evolution, like it's just arose at a different time, I guess. So some things that are different. So the structure's different. Bases are different, right? So there's going to be a different shape to the enzyme because it needs to add ribose versus deoxyribose, right? So there's going to be a different shape. Primer is not required. Not like, remember, in DNA polymerase, you need a primer. RNA polymerase, there's no primer required. It's not as accurate as DNA polymerase, but it has a basic proofreading mechanism. It can back up. So it's moving forward, adding bases as the wrong base. I'm guessing it detects it by a lack of fit because a properly base paired base is going to fit better than an improperly base paired base, but I'm not 100% sure of how it recognizes it, but it's probably that. And then it will back up and remove it by addition of water. So it's like a reverse polymerization reaction. Okay. So the error rate of RNA polymerase is 1 in 1,000. That's actually almost the same as the error rate of reverse transcriptase in HIV. So actually tolerates, so that tells you a higher error rate is tolerated in transcription, not in DNA replication. So why do you think that is? Why would we tolerate a higher error rate when making our RNA versus when we're making our DNA? Yes? Okay, so amino acids have multiple codons. One base pair mistake might have an effect, but that's true also for DNA, right? Yes?
If the DNA copy is messed up, then every one of the mRNAs is going to be messed up, but if you mess with one of the mRNAs, then you have one messed up.
Absolutely. If the DNA sequence gets messed up during replication, your genome is messed up. Every RNA ever made from that gene ever again in the daughter cells will have the error. Whereas if you're making 1,000 RNAs from one gene and one gets messed up, so what? That if it's a mutation that affects protein structure, it'll misfold and be degraded and it'll be gone. Whereas hopefully mostly they'll be okay. And that could also be why so many RNAs are made because of that error rate. Does anyone want to add to this thought? No? Okay. Okay, so I keep telling you there are these non-coding RNAs. So these are the different types of RNAs that are made. A coding RNA, the RNA that encodes a protein, that's what's called coding RNA, is the mRNA, right? So then you've got your ribosomal RNAs, right? So that's what makes your ribosome. tRNAs, that's what transfers the amino acids to the growing polypeptide. Small nuclear RNAs involved in splicing. Small nucleolar RNAs also involved in RNA processing. Small cagal RNAs modify other small RNAs. microRNAs, some of my favorites. They regulate gene expression by blocking translation, and also siRNAs, which also degrade RNAs. And then there's other non-coding RNAs. There's a lot of RNA that never make a protein. So you should acknowledge the fact that it's not all about DNA, RNA, protein. Okay? It's DNA, RNA, something. Okay, any questions about this? Any thoughts? Okay. This usually generates a lot of discussion. Yeah.
I've been hearing MI and siRNAs, but they've been used pretty much interchangeably.
Yes.
So
Yes, so the question is about microRNAs and siRNAs and how they're sometimes used interchangeably. siRNAs, I think it's very clear that they're involved in degradation of specific RNAs that they base pair with. And they are these, short 12 or so nucleotide RNAs, double-stranded RNAs. MicroRNAs are these short hairpins that are processed, I think, sometimes into an siRNA or something with an siRNA-like function. It tends to be longer. And then there's discussion over whether or not microRNAs degrade RNA or whether they inhibit translation by kind of blocking translation machinery. I don't know if the jury's out or if it's been answered, or if there's actually two functions. But they are kind of interchangeably used because they are these small RNAs. They do both function by a base pairing interaction and both prevent expression of a protein in some way. Yeah. Yes?
If an RNA is messed up enough where it actually becomes a different structure, can it be used as a different RNA type, or is it just thrown away?
So if an mRNA
Yeah.
is mutated or something's wrong with it, so it forms a secondary structure and cannot be translated, will be used as something else. So you're saying can an mRNA actually also function as another type of RNA?
Yeah, if it's messed up enough. You know what I mean? Instead of being thrown away, can it be used as whatever?
I've heard about introns being used as microRNAs to regulate that same gene. Heard about it. I can't tell you much more than that. I don't know. I guess that would be smart for the genome to have evolved a way to use something that's messed up, but I think that would be actually, let's think about it for a minute. Okay, so you have an RNA, there's mutations, it forms a crazy secondary structure that looks like a splicing protein, or splicing RNA. Is that a good thing for the cell? That'd probably be really bad because it kind of takes away a level of regulation. Right? I guess it could happen, but would you want it to happen? I like the way you're thinking now. Yeah.
Do you want mutations to happen? No, that's over.
Do you want mutations to happen? Well, yes and no, right? We're at a very low level. For us, if you're a virus, yes. You want less mutations, probably. Yep, so I would say probably it would not, if it was used as something else, it might not be a good thing, because it would be without any regulation. Because this is all regulated, right? Gene expression is highly regulated. Okay, any other questions, thoughts about small RNAs that don't code for protein?
Completely unrelated question. What's the rate of nucleotide addition for DNA replication?
What is the rate of nucleotide addition for DNA replication? I've got no idea off the top of my head.
It's got to be way faster than 20%.
It's got to be really fast because you're looking at a whole genome, but you also have multiple origins. Don't forget, you have multiple origins, so it's not like, yeah. It happens relatively quickly, though, S-phase. Okay. Okay, so let's talk about transcription for a minute. So this is the transcription cycle in bacteria. And then we'll go a little bit more deeply into it on Friday. So basically, it all has to do with recognition of the promoter. So that's how you recognize your transcriptional start site. Got an email from someone. I had that question. I should have inserted it here on the quiz this weekend about you're trying to find all the transcription. Was it you? Who sent that to me? If you're trying to find all the transcriptional units in a genome, and it was a bacterial genome because that makes it simpler, and you were going to create a program to identify them, what would you look for? And the question was, why isn't it ATG? Like, that was the question I got via email. And I said, well, you're not looking for proteins. You're not looking only for mRNAs. You're looking for all transcriptional units. So that includes all of the RNAs. And so it would have to be something that would be on all transcriptional units. And so that's what we talk about when we talk about the promoter. That's a certain sequence of DNA that's recognized. In bacteria, it's recognized by the sigma factor that's going to bind. So the sigma factor binds to RNA polymerase and helps to recruit RNA polymerase to transcriptional start sites, right? Makes sense, right? Because how else is RNA polymerase going to know where to transcribe? If there's no level of regulation, it's just going to bind anywhere on the DNA and start making garbage, right? So you need some way to identify transcriptional start sites. Now, in eukaryotic cells, transcriptional start sites have other things in common, right? Other than just DNA sequence. Who knows something that might be a good way to find a transcriptional start site in eukaryotic cells, other than just a DNA sequence? We'll get to it, but I'm just wondering if anyone's thought about how chromatin structure might affect transcription. So there's certain,
I was going to say, based on methylation.
certain modifications. Histone H3 acetylation, I forget which lysine acetylation, is indicative of a transcriptional start site. And so if you look at the whole genome, and people have done this, where they looked at the whole genome and noted every place where you have high levels of acetylation of a certain glycine, and it's associated with location of transcriptional start sites. In bacteria, obviously, you don't have histones, so it's different, and it's based on the recognition of the promoter by the sigma factor. And so basically, the sigma factor recruits RNA polymerase to the transcriptional start site. The double helix is opened up, and you have this beginning of transcription, which is not efficient at all, and it's called abortive initiation. And it takes a little while to get started. And then once RNA polymerase transcribes about 10 nucleotides, then it starts to move forward, and it kicks off the sigma factor, and then RNA polymerase continues transcription until, and then you see over here, you've got your RNA coming out of this RNA exit, so it's not base pairing with your DNA. And it forms this, at the termination site, it will form a kind of a hairpin structure, which will kick the RNA polymerase off and then release the RNA. Okay, so it's initiation, which is recognition of the promoter. Abortive initiation, so it's kind of slow to start. Once it starts efficiently, it kicks off sigma factor, transcribes the whole gene until it hits the termination site, which is dependent upon an RNA structure, a secondary structure in the form of a hairpin. That's what you're seeing here, this hairpin. And then RNA polymerase gets kicked off and you're done. That's just one copy. Obviously, this is happening many, many times. Okay, I think this might be it for today. Yep. Okay, any questions about this before I let you go? We still have another 8 minutes. Okay, so feel free, those who are taking an exam, to get food because an exam on the empty stomach is no good.